Thalassemia are inherited disorder of globulin synthesis. The basic defect in all types is the absence or reduced output of one or more of the globulin chains. Globulin chain. Pathophysiology: the severity of thalassemia is directly related to the degree of the globulin chain imbalance. The excess globulin chain aggregate, forming inclusion body, premature destruction of the red cells. The secondary complication: bone defect, splenomegaly, endocrine and cardiac damage. Classification: we can classify thalassemia, uh, genetic classification, clinical classification, molecular aspect, explain phenotypic diversity, globulin gene expression, globulin gene switching, pre-implementation diagnosis. Phenotype diversity: clinical severity varies widely, ranging from asymptomatic form to severe or even fetal entities. Phenotypic and genotypic relationship: in alpha thalassemia, it's a straight deletion of one gene, deletion of two genes, carrier, deletion of three genes, a disease, deletion of four genes, HB parts. In beta thalassemia, there is genotypic phenotypic diversity. Mechanism underlying phenotypic diversity of beta thalassemia: heterogeneity and variable severity of beta thalassemia. Beta 2 plus, beta 1 plus, beta 0, dominant inherited beta thalassemia, unusual cause of beta globin deficiency, secondary modifier alpha globin phenotype, variation in fetal hemoglobin production, tertiary modifier loci outside globulin. Heterogeneity of beta thalassemia allele: the most reliable and predictive factor of disease severity is the nature of the beta globin locus. The beta globin locus deletion, rare, point mutation within the beta globin gene or its flanking sequence, mutation outside the beta globin complex, general transcriptional factor, and erythrocyte specific transcription. Deletion: it's rare. Deletion range in size. Apart from this, in the globin gene, the other deletions are the functional and the phenotype interest because they are associated with unusual high HbA2 in the heterozygote. Point mutation classification according to the mechanism by which they affect gene function: transcription, mild to minimum, uneven silent, RNA processing at splicing sites, exon or intron, cryptic splices site, other RNA processing, expression of RNA translation, initiation or extension. More than 30 dominant inherited beta thalassemia allele have now been described and others also discovered. The phenotypic resemble thalassemia intermediate. They have been recorded in the several dispersed geographic regions. It's a majority in single families, many as de novo events, unlike beta recessive form which is prevalent in malaria endemic region. Unusual cause of beta globin deficiency: transposable element occasionally disrupt human genes, result in their inactivation. Secondary modifier: co-inheritance of alpha thalassemia, clinical severity of thalassemia, co-inheritance of extra genes, the level of alpha globulin stabilizer protein. Secondary modifier: variation HbC production, genetic determinants that influence GBF response within beta globulin gene, a complex trans-acting loci mapped to 3 regions of the genome. Tertiary modifier: with increased lifespan of beta thalassemia, subtle variation in the phenotype regarding the complication. This may be affected by genetic variation, hyperbilirubinemia and gallstone, progressive osteoporosis and osteopenia, cardiac complications. Phenotypic diversity is basically determined by the nature of beta globulin gene mutation and modified by other factors. Better characterization of molecular pattern of thalassemia predicts the clinical course without need for a period of observation to determine that the frequency of blood transfusion needed by the patient and hence early better therapeutic decision. Globulin gene expression, globulin gene switch during ontogeny. two switches occur in beta globin gene expression that reflect the changing oxygen requirement of the fetus. The first switch from embryonic to fetal alpha globin occur at 6 weeks of gestation. The second switch from gamma to the beta globin occur or shortly after birth. Post-conception age, and here the postnatal age. Regulation of human fetal hemoglobin switch. Understanding the molecular event in hemoglobin switching will give new approach to the therapy of beta thalassemia and sickle cell diseases. The player at the beta globin locus, thus acting sequence, the genome architecture of the human beta globin locus affects the development of regulation of gene expression. The beta globin LCR integrin sequence, transacting factors, erythrocyte cell specific transcription factor, stage specific transcription factor, chromatin remodeling activities. Genome architecture of human beta globin locus affect the development of regulation of gene expression. Beta globin LCR is required for normal high level globin gene transcription, contain 5 critical DNAs, 1 hypersensitivity sites, which are the site for interaction with transcription factor and downstream gene sequence. In contrast to upper cis-acting element, which functional local, LCR function over long distance to activate globin gene expression. Intergenic sequence, this is the normal, the beta thalassemia and the HFH and thalassemia conform. Transcription factor. Many transcription factors controlling beta globin gene expression have been identified and characterized. These factors form a network of protein and protein DNA interaction with each other. Globin gene promoter, LCR, HSS, and the other cis-acting integrin regions. Transcription factor include erythrocyte specific transcription factor, stage specific activation of beta globin gene, and activation globulin gene, repression of epsilon. Chromatin remodeling. Chromatin exerts control over gene expression by alternating between so-called open and closed conformation or states. Open chromatin is generally DNase sensitive and hyperacetylated, whereas closed chromatin is DNase insensitive and underacetylated. Chromatin remodeling. Acetylation of histone in chromatin leads to expression of specific gene at structure gene loci, including beta globin locus, by making gene sequence more available for transcriptional factor. Deacetylation by histone deacetylase can repress specific gene function at many loci and may be involved in presenting gamma globin gene expression. Biocomplex bind to a base pair polyacrylamide DNA sequence. Chromatin remodeling complex. Complex found only in adult hematopoietic cells. More recently, single complex with acarose as DNA binding subunit and contain both positive acting protein subunit complex that activate gene transcription and represent protein subunit of NER complex contain complex that can repress transcription. The proposed role of biocomplex in human hemoglobin switching and the other chromatin remodeling complex and the transcription factor interaction lead to conformational changes. The complex interaction of the player at the beta globin locus, interaction between the beta and LCR and the downstream gene sequence, transcription factor, integrin sequence, chromatin remodeling element necessary for globin gene expression and globin switching, special combat configuration. Analog model, facilitated tracking model, linking model. Looping model, transcription factor bind to the LCR, the gene promoter. Tracking model, sequence specific transcription factor bind to LCR forming complex, track down DNA sequence. Facilitate tracking model. Aspect of both looping and tracking model are combined. Linking model sequential binding transcription factor along the DNA direct chains in chromatin conformation, and define the transcriptional domain. Conclusion: although many of the events controlling the activity of beta-globin locus are known, the tail of those regulating normal human hemoglobin switching and reactivation of fetal hemoglobin in adult hematopoietic cells remain to be elucidated. If the molecular event in hemoglobin switching or gamma globulin gene reactivation were better understood and fetal hemoglobin could be fully reactivated in adult cells, the insight obtained might lead to a cure for these disorders. Parental diagnosis. Parental diagnosis: fetal blood sampling from 19 to 21 amniotic fluid cells. Recent development in parental diagnosis: the recent development of nested PCR, which can amplify DNA from single cell. Advances in perinatal diagnosis by studying fetal cell in maternal blood. This method provides an attractive, non-invasive approach for parental diagnosis. Pre-implementation diagnosis: to study the oocyst in vitro pre-implementation in uterus, avoid possibility to terminate an affected pregnancy. Nested PCR: increase the sensitivity and specificity of PCR. Two-round amplification: use two sets of amplification primer. Thank you.